working with plants. And in my personal opinion, Paula is one of the best plant scientists in Portugal. She has published amazing work in very high-ranking journals, and she is an EMBO member since 2017. So I'm not just the only one who thinks she's quite a good scientist. So I will shut up now, and we are all looking forward to hearing your talk. And thank you once again, Paula.
Thank you. Thank you, everyone, for joining. Thank you, especially Manuela, for the extremely kind introduction and also for the invitation to participate in this seminar series of yours. Okay, so as Manuela said, I work at the Instituto Gulbenkian de Ciência, also known as the IGC, where I head the plant molecular biology group. It's one of the 3 plant groups there. And the lab is currently composed, besides me, of 5 postdocs. I wish I could, yeah, I think it's working. 5 postdocs. Tom is from Toulouse, Guilmar is from Barcelona, Maria Fernanda, also known as Maf, is from Bogota, Ster is from Coruña, and we also have 5 PhD students. Oops. Alba, who like Ster is a Galician from Coruña, and Rui and Zé, who are from Coimbra, both from Coimbra, and they're both shared PhD students. Zé Pedro is shared with a lab at the VIB, with the Van Berzingam lab, and Rui is a PhD student shared with the Jones lab at the Sainsbury Laboratory. I'm having some difficulties here with my presentation. I had half of my slide covered, and I forgot Dora, who's a postdoc from Kecskemét in Hungary. And we also have Romana, who has been here added to the picture because she is a PhD student from Cuba, and she joined last spring. And unfortunately, we haven't been able to take a proper non-masked photograph since then of the group, hopefully soon. Okay, so what do these people do? To put it in very, very broad terms, what we do is we use Arabidopsis thaliana as a model system to try and understand how plants perceive and then mount responses to environmental signals, particularly adverse environmental cues at the molecular level. And for us, molecular level basically means mRNA splicing, specifically alternative splicing, which is a process that allows the same pre-mRNA or the same gene to generate multiple mature transcripts and potentially mature proteins from the same gene. And we also study a specific group of membrane transporters called the major facilitator superfamily. This is one of the 2 largest groups of transporters on Earth. Together with the ABC, it is also the only 2 groups that are found in all living forms. And as you will see in a while, they have also allowed us to find interesting alternative splicing events. So our study of these membrane transporters has allowed us, using molecular biology, genetics, and also heterologous expression in yeast, has allowed us to find many new transporters in plants with essential functions in the response to stress. For instance, in the root, we have found 2 plasma membrane transporters that are essential for survival in the phosphate starvation. We also found a plasma membrane transporter that is involved in potassium and cesium homeostasis in the plant, or another one, ZIF2, that regulates the response to heavy metals. In the leaf, we have found a potassium transporter that regulates stomatal movements and thereby drought responses. And more recently, we expressed 2 transporters from yeast in Arabidopsis and found that these confer resistance to a broad range of toxic compounds. And so we're still pursuing this line of work with MFS transporters to find out how broad this role in abiotic stress tolerance is for this superfamily of transporters. But as I said, our main focus is on pre-mRNA splicing, which, as I believe everyone knows, is the process through which the non-coding introns, shown here in gray, are removed from the precursor mRNA to generate the mature transcript, which can then be exported to the cytoplasm to be translated into protein. And so it all starts when specific sequences in the introns, called splice sites, are recognized by ribonuclear particles called SNRPs. And the spliceosome, which is one of the largest molecular complex in the cells, assembles on the pre-mRNA. So 5 individual SNRPs, together with a large number of other accessory proteins, assemble on the intron, and upon a series of conformational changes, bring the 5' and the 3' splice site together to form the spliceosome and excise the intron. The intron is released in the form of a lariat. The spliceosome then disassembles so the SNRPs can be reutilized at another intron, and the mature mRNA can leave the nucleus. So one thing I would like to leave clear is that there is a lot of ATP used in splicing, but only in the assembly and the operation of this large complex that the spliceosome is. Splicing itself is a very simple and spontaneous 2-step chemical reaction that requires no energy input. There's a first transistorification reaction with a nucleophilic attack of the branch site adenine to the 5' splice site, and then a second nucleophilic attack from the newly released OH at the 5' site to the 3' splice site, releasing the intron lariat and joining the 2 introns. Okay, so our focus is more specifically on alternative splicing. As I said, the process that allows the same pre-mRNA to be spliced into more than one different way, generating more than one transcript and potentially more than one protein from the same gene. And this can happen in a variety of ways, such as in an exon skipping event that I just showed you, but it can also result from the selection of alternative 3' or 5' splice sites, which result in exons of different lengths. There are also cases of mutually exclusive exons when either only one or another exon are kept in the final RNA, or particularly in plants, this is a very common alternative splicing event, intron retention, cases where specific introns are retained in the mRNA. And so alternative splicing is very important basically to generate proteomic diversity. It greatly expands the coding capacity of a genome. And it can also be important to regulate gene expression, either by differentially splicing parts of the transcript that are important for their stability, or more often by changing the frame and introducing premature stop codons that then target this transcript for degradation by a process called nonsense mediated decay. So as I said, the splicing process depends on the recognition of specific sequences in the introns. In more than 99% of introns in all species that contain introns, these sequences start with a GU and end with an AG. The rest of the 5' and the 3' splice site are very degenerate. So the consensus is actually not very strong. And only an A defines the branch site. And as I said, the SNRPs recognize these sequences in the intron to assemble a spliceosome and execute the excision of the intron. But because these splice sites are short and degenerate, they're usually not sufficient to drive the assembly of a functional spliceosome. There are other cis elements in the pre-mRNA, such as intronic or exonic splicing enhancers, which are bound by accessory proteins of the spliceosome, such as SR proteins, that have an RNA recognition motif that recognizes the RNA and an Arginine serine-rich domain that is involved in protein-protein interactions. And what it does is it recruits the SNRPs, the core components of the spliceosome, to the nearby splice sites. So the strength of a splice site depends not only on its consensus sequences, but on the concentration, the amounts and activity of different accessory proteins, such as SR proteins, that are present in a given cellular context. And so alternative splicing is known to be a very prevalent feature of eukaryotes. Bacteria don't have introns, but higher eukaryotes are extensively, the genomes of higher eukaryotes are extensively alternatively spliced. So in our genome, it is known that more than 95% of the genes that contain introns produce more than one transcript via this mechanism. And in animal systems, alternative splicing is known to determine key biological processes. It is the alternative splicing of a specific gene called sex lethal that determines the sex of the fruit fly. Alternative splicing of a potassium channel in the inner ear determines the recognition of different sound frequencies. A very interesting paper almost a decade ago found that alternative splicing can change a temperature sensor in the nose of bats and place it somewhere else in the brain to detect lower temperatures and thereby check for hot spots, places where the blood flows at the surface of their prey. And of course, alternative splicing is involved in many human diseases, many cancers, Alzheimer's, cystic fibrosis, to name just a few, are diseases caused by mutations either in splice sites or in spliceosome components, okay? And when I started the lab 12 or 13 years ago, the idea was exactly to explore the functional relevance, the biological meaning of alternative splicing in plants, because it's really, it's still not as established as it is in animals. We know now it plays an important role in the response to stress, as I will show you soon, but it is still very poorly understood, the relevance of alternative splicing when compared to animal systems. And we have been approaching this question in two very different ways. Initially, we started by trying to identify individual genes in our favorite model, in Arabidopsis, where an alternative splicing event would have clear biological meaning. So we started by studying individual case studies. But when we'll see later in my talk, we are also using a more broad approach by doing the functional analysis of SR proteins, which are the established regulators of alternative splicing in mammals and have highly conserved counterparts in plants. So the idea is that by studying a family that regulates, that controls alternative splicing, we will understand the functional meaning of this process in plants. And so starting by our first approach, I want to give you 2 or 3 examples of alternative splicing events that we found and reported have clear biological impact in plants. One of the first papers in the lab studied an exon skipping event, Exon 8, in a ubiquitin ligase gene. And we found that this exon specifically encoded a nuclear localization signal. And this determined the targeting of two isoforms to different places of the cell, while the one that retained exon 8 was in the nucleus, the one that excluded exon 8 was found in the cytoplasm. And this in turn dictated had a differential impact on the formation of the apical hook, which is this structure that protects the apical meristem, where the stem cells of the plant are, during growth through the soil. And then, as I told you, as I hinted at earlier, our work with MFS transporters also revealed interesting stories where alternative splicing had a striking impact in the biology of the plant. So in ZIFL2, we found an alternative 3' splice site selection event that resulted in 2 transcripts, 2 mRNAs that differed in only 2 nucleotides. And this, because this changed the frame, this resulted in the introduction of a premature stop codon in one of the transcripts, generating a truncated transcript, and a truncated, not a truncated transcript, a truncated transporter. And what we found was that the full length and the truncated version were having not only different subcellular localizations, the full length was at the tonoplast and the vacuolar membrane, the truncated version was at the plasma membrane, but they were also in different tissues in the plant. One was functioning in root development because it affects polar auxin transport, and the other one was in the guard cells regulating stomatal closure and then by tolerance to drought. So one gene was fulfilling 2 very different roles thanks to alternative splicing. And yet another transporter that we found was a zinc transporter conferring tolerance to zinc, to the heavy metal. We found that an intron retention event in the 5' UTR, so in the untranslated region of the gene, although this alternative splicing event did not change the protein, it was allowing the protein to be more actively translated, more efficiently translated. So it was a zinc-regulated alternative splicing event that produces more protein and thereby promotes plant zinc tolerance. But what I really would like to tell you a bit more today is about our functional analysis of SR proteins. As I told you before, SR proteins are the key regulator of alternative splicing. They not only interact, recruit, and stabilize spliceosomal components at nearby splice sites, promoting splicing. They can also antagonize the activity of splicing silencers. And they form a network of protein-protein interactions across the intron, pulling together, bringing together the 5' and 3' splice sites for the splicing reaction to occur. So because they are key factors in the recognition of splice sites, because they can influence splice site selection in not only a concentration, but a phosphorylation dependent manner, because these proteins are heavily activated by phosphorylation, at least in mammals. And because they have been found to be highly conserved in all genomes that undergo alternative splicing, SR proteins are widely recognized as the major regulators of alternative splicing. And as such, it is expectable that analyzing their loss of function can give us key insight into the biological functions of alternative splicing. And that's exactly what we have been doing. The Arabidopsis thaliana genome codes for 20 different SR proteins. This family is largely expanded in plants. We have only 12 SR proteins. Arabidopsis has 20. And what we have been doing is we have been generating loss of function mutants and also overexpression lines for these individual SR proteins. And we have established very well the role of two of them that I will briefly tell you about, but we have also recently found phenotypes for many other of these genes. And what we have been finding is that they play a key role in the ABA pathway. And as you may all know, abscisic acid is one of the 5 classical phytohormones. It plays a very important role in development, particularly in the seed, where it promotes desiccation tolerance, the establishment of dormancy, the storage of nutrients, and it also is a potent inhibitor of germination. But ABA is perhaps better known for its role in the response to environmental stress, particularly stresses that cause osmotic stress to cells. And although plants are also known to possess, to a certain extent, ABA-independent response mechanisms to these forms of stress, an adequate response to drought and salt, but we know also temperature stress now, requires a huge increase in the endogenous levels of ABA. So ABA is massively produced in cells in response to osmotic stress. And this then leads to downstream signaling, namely to the de-repression of these key kinases that then will phosphorylate either ion channels, which leads to rapid closure of stomata to confer stress tolerance, but also phosphorylation of transcription factors that induce a wide reprogramming of the transcriptome to promote stress tolerance. This involves the synthesis of dehydrins, osmolytes, and so on, protective genes, but also induces during early plant growth, germination, and the transition to chlorophyll, to greening states, a heavy growth arrest to protect the seed or the seedling until more favorable conditions arise. So because of this massive transcription reprogramming and because of this key phosphorylation events, the ABA pathway has in the past been extensively studied at the transcriptional and the post-translational levels, but very little is known of the regulation of this pathway at the post-transcriptional level, at the mRNA level, okay? And so coming to our specific study of SR proteins, coming back to that, the SR protein that we have longest studied in the lab is SR45. This study started with the PhD project of Raquel Carvalho. And what she found in 2010 was that SR45 was essential for the response to exogenous sugars. So it regulates sugar signaling in the plant. So under moderate concentrations of glucose that are not enough to inhibit the growth of the wild type, the SR45 loss of function mutant showed a clear growth arrest and was unable to expand and green these embryonic leaves, the cotyledons. And we knew this was specific to sugar sensing because if we used equimolar concentrations of mannitol, osmotic pressure, we saw no difference in the development of the two genotypes, indicating that what we're seeing is specific to sugars and not an osmotic stress response. And because sugar signaling is known to require ABA production, we then used an inhibitor of ABA biosynthesis called fluridone and found that our phenotype is partially rescued by this inhibitor, indicating that this growth arrest is at least operating via the production of ABA. And so we tested this by checking the endogenous levels of ABA in 3 genotypes. We used the ABA2 biosynthesis mutant enabled to respond to ABA to stress by producing ABA. And we found that indeed our mutant responds to glucose by producing much more ABA than the wild type. And then regarding the second oldest story, SR protein story in the lab, which was the PhD of Sofia Carvalho and is now being followed up by Tom in the lab. What Sofia found initially is that knocking out SCL30A generates plants that cannot produce big enough seeds. So the seeds are smaller. And we immediately asked whether they had problems germinating, and they do not have a significant effect in germination. After 2 days, the seeds are fully germinated like the wild type. However, if we put these seeds germinating under salt or drought stress, this mannitol high osmotic pressure mimics drought stress. Although we see no difference in optimal conditions, under salt and drought stress, there is a massive inhibition of the ability of the mutant to germinate, indicating that this gene is required for germination under stress. And the next question was again whether this was ABA dependent, and it's almost fully ABA dependent, because if we look at this phenotype in the presence of fluoridone, we've rescued the ABA defect. So the control that SCL30A exerts over osmotic stress during germination responses requires the endogenous accumulation of ABA. And another interesting thing we know is that plants overexpressing this SR protein show the opposite phenotype of the mutant. They produce larger seeds and can germinate better under salt and drought stress, which has obvious potential biotechnological applications. Okay, so to summarize, we have extensively looked into 2 SR proteins, SR45, which negatively regulates sugar sensing during early seedling development by repressing the ABA pathway. And we also found that SCL30A negatively regulates seed size and ABA-dependent osmotic stress responses during germination of the seed. So although these 2 SR proteins clearly respond to different external signals, to different stresses, they respond to different exogenous cues, they both converge on downregulating the ABA pathway. And in doing so, they might, under moderate conditions, allow seed germination and growth and promote tolerance to the stress. Okay, so I have told you about 2 proteins, that 2 splicing factors that we have extensively studied, but let me tell you what we have been finding in the last year or 2. So unlike in animals, plant development is post-embryonic, meaning that all the development happens after the plant is born. So when a buried seed germinates, the first event is the protrusion of the embryonic root, the radicle that grows downwards. And the next step is the emergence of the hypocotyl, the embryonic stem, which grows very rapidly in the dark. This is an astonishing rate. The hypocotyl can, elongate the cells like 15-fold in just 48 hours to rapidly reach the light. And when the hypocotyl reach the light, the embryonic leaves, the cotyledons, quickly expand and turn green, allowing the plant to transition to the autotrophic phase, where it can photosynthesize and make its own food. And these 3 early steps of plant development, of plant growth, are not only essential and crucial for the viability of a plant, but they're also particularly sensitive to stress, and ABA plays a crucial role at these 3 stages. And what we have been finding lately as we go ahead with our individual analysis, functional analysis of SR genes, is that not only SCL30A and SR45, which I've told you about, that regulate seed germination and cotyledon development, but virtually every other SR protein that we look at plays a role in regulating stress responses via the ABA pathway in one or more than these 3 stages. And some of them are specific to an organ, to the seed or the hypocotyl or the cotyledon, and that's where they work, others not. And curiously, there are others that repress the stress response, the ABA pathway, the ABA signaling, and others activate ABA signaling. Another striking thing is that these genes seem to be ubiquitously expressed in the plant and are not significantly regulated by ABA. So this indicates that the mRNAs are there and maybe the proteins are there, but they are somehow activated specifically at these stages in opposite regulatory mode when the stress appears. Okay, so this leads to a number of very simple conclusions. First of all, one thing it's clear from these results is that the ABA pathway is not only regulated at the transcriptional or the post-translational level, there is also a clear and novel RNA layer of regulation that needs to be explored. This SR protein mediated of the control of ABA responses plays a prominent role specifically during these first stages of plant growth, because our mutants and overexpression lines do not seem to show evident, at least, phenotypes at other stages of development. And this indicates that alternative splicing is likely to be exerting a broad control of plant stress tolerance during early plant growth by targeting ABA signaling components. And that is now our challenge, to identify the mRNAs that these SR proteins are acting on to control stress responses in an ABA dependent manner. And we are going to use an iClip. We are already using iClip, which cross-links with UV proteins with RNA and allow us to pull down tagged SR proteins in vivo from the plant to look at which are the specific mRNA molecules that are bound to our SR protein under the stress conditions where we see the phenotype. We will then need to do RNA-seq to see what the effect of the loss of our SR protein is on these direct target mRNA, but also the broader effects in the transcriptome that this has. These mRNA targets that we identify will, of course, to be then functionally validated. The idea is that we need to replace the defective mRNA pattern, splicing or expression pattern in the ABA, in the SR mutants, and see that we can recover the ABA defects by reestablishing the correct pattern of RNA expression or splicing. But we are also interested in the upstream regulators. As I said, ABA signaling is massively regulated by phosphorylation events. So SNRK2 kinases phosphorylate ion channels, transcription factors, but we believe that there are also phosphorylating splicing factors to regulate the ABA pathway at the mRNA level. And so we need to find not only the interactors, but the phosphorylation events. And we're planning to do this through mass spectrometry. And these events and interactors will then need also to be functionally validated by genetics. And this is what I wanted to tell you today. I'm sorry, Manuel, I think I took longer than I should. You had told me 30 minutes. I hope it was not too bad. Just let me finish by thanking again the lab, the 10 plant molecular biology lab members who are great fun. They're not also only very dedicated scientists. I can see in the last few months how their dedication to science in extremely difficult conditions has been, but they're also truly nice people and fun to interact with. The work I showed had also, lots of participation of former lab members. These are all collaborators. Funding comes from all these different sources. And of course, I'd like to thank Manuela again for the invitation and everyone out there for listening. I'll be happy to take questions if anyone has them.
Thank you, Paula, for this amazing talk. Really nice to see cool science. And I hope some of our students or people in the department have questions for you. So I'm not sure, Andrea, if you're there.
Yes.
Okay. So do they have permission to talk?
They are allowed to turn on the cameras and their mic if they want.
Okay, so do they have any questions?
I have a question. Oh, I can hear myself. Wait a second.
Not too much.
Sorry, we're in the lab and we've got a sound thing set up so we can hear it, and it started doing a feedback loop.
But we're hearing you well now, Tomas. You can go ahead.
Anyways, so one of my questions was more a practical question from the beginning. It was, how do you go about looking for important splicing events? Like, how do you find cases where splicing events are important for the plant?
Okay, that's a good question. So nowadays it's fairly easy, because in the last, I would say 5-6 years, you go to databases where the genome is annotated and you can see the prediction of several different transcripts for the same gene, okay? But that was not so easy 10 years ago when I started the lab. So what I would do is, if I were interested in one specific gene for some reason, I would just design primers at the UTRs of the gene, so at the very ends of the RNAs, and I would just, use lots of cycles in different tissues under different conditions and see if I could find different bands. Of course, you have to sequence the bands because there's many reasons why you can see weird bands in PCRs. But and then I started realizing that the annotation 10 years ago had to be very incipient, because every gene I tested had lots of splice variants. So nowadays, any gene you want to study, you can study splicing, because the chances of finding a only constitutive splice gene is very low. If you don't see it, I can tell you, if you go to different tissues or to different environmental conditions, you will see a different transcript.
So you had to look at specific genes that you already had in mind when you found.
Look, I was interested. So when I first started with this idea, I was in the Chua lab at Rockefeller, in one of the 2 labs that Manuela said I worked. And so I had done a postdoc on ABA and seed germination in the plant lab. And then by chance, I ended up leaving plants and doing splicing in mammals. And then when I had the opportunity to come back to Portugal, I wanted to do splicing in plants because very few people were doing it. And so I came back to the lab and I said, what about the genes I had been studying for ABA response? And I picked those and I designed these primers, and I was seeing different bands, especially when I used lots of cycles, because you end up amplifying lots of things. And then I sent it for sequencing, and all of them had splice variants.
Okay. Well, I've got one more question. Well, I've got a few more, but I've got one more that I would like to ask, if that's okay.
That's more than okay. Thanks for the question.
You on the example with the where the gene had a difference in only 2 nucleotides.
Yes.
And it ended up truncated. Was the difference that they had different nucleotides, or that they lost 2 nucleotides?
It's a 3' splice site event. Let me try and use this to show you. Okay. So remember here.
Yes.
Okay. So the difference in splicing, the 2 versions, was a alternative 3' splice site, which means the spliceosome always recognized this 5' splice site. And then instead of always cutting the intron here, recognizing this 3' splice site, it would sometimes recognize one or the other. And the difference here, instead of being this big chunk, was like an A and a T.
Oh, okay.
And so one transcript would have minus an A and a T here, and the other would have the A and the T. So the
And
full
with this,
thing was exactly the same, except for different for 2 nucleotides. It was actually very difficult for us to find these 2 transcripts, because by PCR, it's very difficult to identify them. And what this made was, you know, if you don't take, if this difference here is not 3 or a multiple of 3, you will change the frame of the protein downstream from here, right?
Exactly.
And when you do that, you very often what happens is that you introduce a premature stop codon, and that's how the difference in 2 nucleotides generated a much shorter protein.
Okay. Okay.
And then we were super surprised to see that the shorter protein was going to the plasma, was no longer staying in the vacuum membrane. It was going to the plasma membrane. That's likely because the last part of the protein had a vacuolar signal, right? That was started.
Yeah. Okay, I see. Cool. Very
Thanks
interesting.
for the question.
Thank you.
Any more? I have a question, Paula. So in Arabidopsis, you say you have 20 SR different proteins, and they belong to different classes, yeah?
Different subfamilies, yeah.
And what about during evolution? Do you have fewer of these classes back then, or?
It's interesting. I have Zé Pedro, the PhD student from Coimbra, is for the first time, you know, I had this. He actually didn't want to come to my lab. He wanted to go to Jörg Becker's lab to do evolution, but Jörg Becker could not take him. And so I said, okay, come on, let's find something that Jörg can help us with. And you did a PhD in my lab, and we collaborate with Jörg. So what is Zé Pedro doing? He is looking at SR proteins for the first time in Fiscomitrella in moss, okay? And what he finds is 16 instead of 20. Actually, I'm not even right. He finds, when I say 20, I'm not being exactly accurate. They're 18 because we have these 2 SR-like, which I have put them in the bunch of 20. And what Zé Pedro finds, and has already published in Frontiers in Plant Science a year ago, is that Fiscomitrella has all these 6 subfamilies, and instead of 18, there's 16, so not much of a difference. And then apparently he's finding 3 instead of 2 SR-like, so the difference is not big. So during this transition from water to land, plants either already had or acquired. So we're now trying to look at Mercantia and what is the other, Chlamydomonas, and try and look at the genomes for SR protein. So the question now is, do you already have these stress functions? But I mean, since these early land plants already had introns, you're bound to need SR proteins because they're required also for constitutive splicing.
Okay. And you're trying to relate those to stress responses as well, or not?
Exactly. So Zé Pedro is not being very lucky. He's not yet, after a year and a half, able to generate this Fisco mutant for an ortholog of this SRZ. I think it's this one, which was the one he chose initially. And now we're actually having to shorten our path. And what he's going to do is he's going to try and use his clone to complement the phenotypes in Arabidopsis. So the question is, do these SR proteins in Fisco that he retrieved from an extensive in silico analysis, can we functionally analyze them and try and see if they have similar stress functions, namely an ABA, which is already important in the moss. And so he has been unable so far to generate the mutant, and now what we're trying to shortcut to try and answer if there's any functional conservation is by trying to complement with a Fisco SR protein, the Arabidopsis ABA phenotype.
Okay. Any more questions? So I have another quiz. I have many questions, but So one thing is quite surprising is that you say that the expression levels are a little bit conserved, and you think it's more to do with the regulation of the protein itself rather than the expression levels?
This is not a absolute rule, Manuela. What we do see, I can tell you, SR45 is not induced by ABA, is not changed by sugars. SCL38 doesn't have its mRNA levels changed by ABA either. We actually also published a paper 5 or 6 years ago with this regulation, ABA regulation of all the family. And we find 7 of the 20 are mildly changed by ABA and are differentially expressed in ABA mutants, but it's not significant. So we think it could also be at the splicing level that this regulation is happening, because SR proteins are themselves, it's a general feature of splicing factors actually in any eukaryote. These genes are themselves extensively alternatively spliced. So it could also be that ABA regulates the splicing pattern of these things, but since SR proteins are, established phosphoproteins with many kinases identified in mammals, our guess is that, the proteins are probably there and activated by phosphorylation when stress in ABA kicks in.
Okay. So if there's no more questions, we think we can finish. Thank you very much, Paula, for
Thank you very
being
much
with us.
for the invitation.
No, it was fantastic. It was really great. Hope to see you soon sometime.
Yes, hug you soon. I hope to hug you soon.
Stay well and safe, okay? You and your family.
Yeah, you too. Have a nice lunch, everybody. Thanks for coming.
Bye-bye.
Bye.